2014, we are rewired. And this year, for our foundational advance, we've designed a system which we think can revolutionize biotechnology, allowing it to harness the power of mixed microbial populations so that it can make high-value products that it could never make sustainably before. So what do we mean by this? Well, to answer it, let's take a step back and just have a look at biotechnology that currently operates in a very simple way. So, in biotechnology, we make compounds, and in our project we're focusing on high-value compounds, things like medicinal drugs. You can see cortisone there. We make these in bioreactors using different microorganisms, and we have to feed these things raw materials. Now, at present, the industry largely relies on things like sugarcane and even petrochemicals, things which aren't sustainable in the long term and which could be used for something else, food production, for example. What the industry really needs is a way that it can transition to using so-called waste materials, things like cellulosic biomass, hay, wood shavings there. This would be much more sustainable in the long term. But the problem with this kind of system is that the reactions involved in doing this are so complex, and the starting materials are so varied in their composition, that you can't just have one strain doing this. Even a synthetic strain with lots of parts crammed into it, it's just not enough. What you need is lots of different strains, all acting together, each doing a designated job, a mixed microbial population. So you might think, well, fantastic, problem solved, let's just put all these different strains in one reactor, let them get on with it. But it's not as simple as that. These different strains that we would rely on are not adapted to one another, and they're not going to get on well with each other. So if you put all these different strains together, some of them are going to grow faster than others, some of them are going to be actively harmful to one another, and sooner or later, one of the strains will disappear from the reactor, the whole system will crash, and you'll have to start again. Now, this isn't a profitable way to make these things, and in the industry, if something isn't profitable, it might as well be impossible. And that's where we come in. So we looked at this system, and we realized that if we could make these different strains that the industry relies on communicate with each other, if we could rewire them so that they can regulate their growth according to each other's population, then we could facilitate a system like the one we just mentioned. You could have a system where you have one reactor that you feed it raw materials, and it produces something like cortisone from the other end. So that's what we set out to do in our project. So, to this end, we designed a test system using just 3 strains to demonstrate that it was possible for these strains to regulate their population against each other. And we also modeled this to get the key parameters and check that it was feasible. For the communication, we looked at a relatively new system called mutable wiring that we'll explain later to connect these cells together. And we also characterized some of the dead run tags that were out there as well to make the whole system more responsive. And finally, we took a step back and we wanted to see whether the engineering principles that we built into this were also present in the human equivalent of these systems, iGEM tools. Now, before I hand you over to Philip to discuss the technical details, it's worth looking at this 3-strain system that I mentioned, which we designed. So, it's 3 strains, as I said, and what we wanted was a system where each strain would grow in concert with the others, so no one strain would get too far ahead in terms of population. The way this would work is each strain is emitting a signal, and each strain is also listening out for the signals that the others produce. If they detect both the signals, that's fine, they grow as normal. But if they stop detecting one of them, they turn on their growth regulatory genes and then pause their growth to let the others catch up. Now, you can see in this diagram that we've represented these as logic gates, AND gates. And that's because that's exactly how this works. It's a biological AND gate that underpins this whole system. And the schematic for this is this. So you've got 2 promoters here, which is sensitive for the 2 signals from the other strains. And when these are both active, these create a transcription factor, which activates this repressor, which represses the inhibition of growth, if you follow. So if one of these 2 signals is missing, then this transcription factor will not be present, the repression will not happen, and they will pause their growth because they're activating these growth regulatory genes. Now, that sounds fine, but we realized when we looked at the signaling mechanism that we were going to have to use to connect these, that the traditional method of doing this in iGEM and synthetic biology, quorum sensing, wasn't really going to be suitable for us for some reasons which we'll go into shortly. So we had a look around, and we came across this cool communication system that was invented last year in a Spanish lab by Victor De Lorenzo called metabolic wiring. Now, this is what we eventually went with our project. So I'm going to hand you over to Philip. He's going to tell you a little bit more about this system and some of the other cool stuff that we designed along the way.
Thank you, Sam. So, I'd like to briefly explain the concept of metabolic wiring to you. It is based on the the principle that many biodegradation pathways in nature are performed in steps. And the way this works is by substrate-specific promoters. So you can see in this example, in this metabolic pathway, the first metabolite will act on the first promoter, which controls the enzymes for the first transformation, which will convert into the second metabolite, and so on and so forth. So here you have this hypothetical metabolic pathway that we would like to use for metabolic wiring. And you can see how each metabolite activates the relevant step in the pathway. So if you separate these steps into different cells, effectively what you have is a novel intercellular unidirectional signaling method. And you can see how the metabolites flow from cell to cell. Now, what we've done here is we've fused the last promoter of the pathway with GFP. And what this means is that when the last cell in the pathway receives the signal from the previous cell, it will start to glow. Now, this is just an example to illustrate that you can fuse these promoters with any gene you like, which enables you to design novel intercellular genetic circuits. There are many advantages of this novel signaling method over previous signaling methods. First of all, it is orthogonal to native systems, which means that there is little, if any, interference with the cellular mechanisms. Secondly, there are many more molecules that you can use in parallel with one another when you're designing your intercellular genetic circuits. And finally, as the metabolites are being chemically transformed from step to step, there is no chance for saturation of the signal in the medium, which would normally lead to false firing. So what does this mean? We can now expand our genetic circuits from one cell to many. And this is important because it means that we're no longer confined by what can be fitted in one cell. Instead, we can make use of specialized strains which can all work together in concert. And this is the future of synthetic biology. Now we can control whole communities of mixed microbial populations to tackle many challenges such as the ones that we mentioned earlier. So, back to our system. We worked on 3 wires for wiring up our 3-cell system. The first one, the transgenic wire, you can see here we've taken genes from the biodegradation pathway of Yuma Shino. We've split the pathway into 2 cells. We put the first genes into one cell and the rest of the genes into the other. And you can see this way, the first cell can sense Yuma in the medium, convert it to benzoic acid, which in turn will signal to the second cell to perform a specific function. Similarly, for a second metabolic wire, this genic wire, it relies on the same principles, but instead of introducing heterologous genes into the different strains, we did something different and started from a native pathway, which exists in E. coli, and knocked out 2 different halves. So you can see effectively we split the pathway into 2 different cells. Finally, we have the wood sugar wire, which relies on the carbon hierarchy principles. Here, the first cell signals to the second cell by modulating the ratio of arabinose to xylose in the medium. So after we'd identified the 3 metabolic wires that we wanted to work with, we started looking for genes that will allow us to modulate the growth rate of our exponentially growing populations. We had 2 approaches. The first one was to overexpress a growth-limiting protein such as Lilley, which is responsible for inducing the stress response in bacteria, and has been shown in the past that when induced, it can induce a transient stationary phase, as you can see in the top right corner. So as you can see from this diagram, if not both signaling molecules are at high enough concentration, then the repressor, which is suppressing the expression of Lilley, is not being expressed, which means that the cell will enter this transient stationary phase, effectively waiting for the other strains to catch up. The second method works in the similar principle, but instead of overexpressing a protein, we are suppressing the expression of a gene which is normally required for exponential growth, such as RPOD, the sigma-70 subunit of RNA polymerase. And in this case, we're doing so by acting on the transcriptional level. So we employed an Anderson's RNA method for silencing the transcript. And the protocol that we followed suggested the use of paired termini for increased stability. So you can see there the hairpin loop, which stabilizes the Anderson's RNA. And at the top, you can see the actual Anderson segment, which binds with the target transcript and inhibits the ribosomes from translating the protein. Now, what we found was that when we were generating those Anderson sequences, often the secondary structure would show that there is folding at the binding region, which would probably inhibit the silencing efficacy. So what we did is design a software that would generate all the possible candidate sequences for silencing a particular transcript, and we'll feed them all into an existing program called VNRNA, which will predict the structures. And then our program will take the output and rank it according to the secondary structure in such a way so that we could eventually pick the optimum and this sends RNA sequence for our silencing. Here you can see how the binding region at the top is not folded up. And this was part of our collaboration with Team UCL, who used our optimized transcripts. I'd like to now pass you over to Charlotte, who will explain to you how we proceed with our system.
Thank you, Philip. So, having completed the design of our system, we decided to model it to see if this predicted any problems. In order to do this, we modelled the population of 3 strains, each containing our synthetic system, using ordinary differential equations. The results are shown here. As you can see, the 2 faster growing strains are moderating their growth to the slower growing red strain by alternating between patterns of growth and rest. This is markedly different from the control, shown here, where the red strains rapidly outgrow. So, what do we learn from looking closer at our model? First thing we found was that the level of signal required to permit the growth in each strain was dependent on the sensitivity of the substrate-specific promoters to that signal. In order for the system to work, faster growing strains had to have more sensitive promoters so that their neighbouring populations, which had reduced capacity to produce signal, could still allow them to permit growth. In order to do this in vivo, we'd need to test each promoter with a series of RBSs to see to what degree this tuned the sensitivity. The second thing we found was that the rate of reduction of signal and rate of degradation of signal had to be precisely balanced. This is because metabolic wiring, unlike quorum sensing, degrades the signal when it's received. This is useful as it reduces the probability of signal saturation in the medium, but also makes it possible that if the degradation rate is too high, then the signal is degraded before it can act on the receiver's promoter. So in order to counteract this, the production rate and the degradation, the production rate must be twice the rate of the degradation rate in both receivers. To illustrate this, there's a short animation. Here we can see what happens if the production rate is too slow. Here we can see what happens if the medium is saturated because the production rate is too high. And here is everything working wonderfully together, and all the populations grow together in concert. So, how can we moderate these rates of degradation and production? In order to do this, we could artificially limit the enzymes required for metabolic wiring in each cell, and for that we would need degrons. A degron is a type of protein tag that can be fused to a gene of interest in order to target it to the cellular proteasome. The balance between the production of the tagged protein and its degradation within the cell acts to limit its concentration, as different degrons can provide different degradation rates. In order to characterize some of the degrons from the registry, we decided to fuse them to GFP and RFP and measure their peak fluorescence. Our results for these are shown here. As you can see, we achieved different fluorescences for RFP and GFP overall, but the patterns of the degrons are similar, suggesting that we performed a qualitative analysis to see to assess their strengths. But we had a further use for these degron-tagged fluorescent proteins. We decided to use them as reporters for our system, so that an engineer using our synthetic equilibrium within a bioreactor could assess its status. As temperatures can fluctuate within a bioreactor, we decided to assess the half-lives of degron-tagged GFP and RFP across a range of temperatures. The results for this, for 32 degrees and 37 degrees, are shown here. As you can see, again, the degrons show a similar pattern of strength, suggesting that we have an accurate picture of to what effect they turn over GFP and RFP. So, having finished the analysis of our system, we wish to look at it in a wider context, and Yuma can tell you more about this.
So, for our policy and practices, we first looked at this concept of division of labor, which is what we're trying to do, divide up tasks among different strains of bacteria in order to make more complicated tasks possible. And we looked at the organization of systems, first within our own iGEM team, and then through theoretical and practical examples and a 16-page report. And we came with 5 themes that seemed important for the design of a system. We used these same 5 themes then to construct interviews and question other iGEM teams to see how they thought they should be